Okay, thank you very much. Another reason why I'm speaking about zooplankton here is that most of my research revolved one other way around zooplankton all the time, and I'm also responsible for zooplankton monitoring in the Swedish national monitoring, and I'm also a chair of zooplankton expert network in the Baltic Sea. So I'm kind of now feeling like a spider in a net about zooplankton, and some of these duties I really don't know how to deal with. But here I will try to cover all zooplankton stories in the or all aspects of zooplankton research in the Baltic, starting from how we collect it, what do we do with it, what kind of information do we get from zooplankton samples, how we use this information, and it all kind of listed here. So the ecological classification of zooplankton in general, with particular reference to the Baltic zooplankton, it's a bit special compared to other oceanic or marine zooplankton. It's also special compared to the traditional limnological way of classifying zooplankton and working with it. So we are in between, and this in between makes it more difficult but also more fun. Sampling and analysis, diversity of Baltic Sea zooplankton, spatial and seasonal variability, long-term trends in zooplankton, mostly based on Swedish data. Zooplankton in the food web, again, what does it do and why? We are interested in that. Invasive zooplankton species, how they affect Baltic pelagic food webs. And finally, a little historical excursus into the Swedish zooplankton research from 60s up to date, or not today, but like a couple of years back, and how it was developing both conceptually and methodologically. So why zooplankton is important, I think we all know by for everybody who came here kind of have a guess why. Zooplankton is sitting in the middle of the food web. The transfer of primary production to fish is only possible through zooplankton link. This is the red arrow. Whatever is synthesized in form of organic biomass in the sea can be transferred to fish as a top consumers only via zooplankton. Even if these fish as adults don't feed on zooplankton, they feed as larvae. There are some exceptions. So some zooplankton species can actually feed on dissolved organic material by absorbance. But this, compared to the traditional pathway through feeding on phytoplankton, is kind of very, very minor. So why it's important we know how we classify it. Here it's a little bit of compromise between understanding ecology of zooplankton and logistics of our methods and gears, how we can catch them in the best possible way. Generally, if we'll think about zooplankton in general, both oceanic and limnetic, we can classify it into three major categories according to life cycle. It's holoplankton, species that live as plankton whole life, or meroplankton. Those are larvae of benthic animals that spend only part of their life cycle as plankton. In some areas, they can be really important in terms of the biomass, in terms of the contribution to the productivity, in terms of the contribution to fish feeding also. So this meroplankton is essentially a part of what we sample as true holoplanktonic part of the community. In terms of the vertical position, so our sampling is also kind of restricted to certain vertical part of the water column. In terms of the vertical position, zooplankton is also classified as Newstone, those that are associated with water film, essentially, a few centimeters below the water film. Some of them are feeding on ciliates that are attached like this to the water film, looking down. Some of them are feeding on phytoplankton species that occur as a very thin layer just beneath the water film. So those species are classified as neustone. And using our current and most common sampling gear, we have only a little part of it, not representative part of it, or rather representative, but not, we cannot describe this part using the samples that grab a substantial part of the water column and therefore dilute contribution of this species. Epipelagic and mesopelagic species occur within the trophogenic level layer and below the trophogenic layer. So this is what we commonly sample in the Baltic Sea, and this is what our mesozooplankton samples consist of. In the oceanic areas, where we have substantial depths, like more than 500, 1,000 meters, then we have bathypelagic and abyssopelagic species. Those are several kilometers down there. We don't have them here. We don't deal with them. But in general, it's good to understand that there is a vertical zonation of zooplankton, and when you read literature, when you read papers, research papers describing oceanic plankton, think where it comes from and how comparable it essentially could be with our more or less upper part of the water column species. And finally, we have the marine, which is oceanic, neritic, and brackish water plankton, and we have limnetic or freshwater plankton in general classification again. And here we are mostly having have the brackish water plankton in the most part of the Baltic Sea. Up north, we have a fair contribution of true freshwater species like Daphnia, for example. Down in the Kattegat area, we have a contribution of oceanic species like decapod larvae or decapods, marine decapods. But generally, like volume-wise, it's mostly brackish water species that are here. Size-wise, that's another limitation or another compromise of ecology and sampling logistics. If we split all the plankton categories into size, then we have usually refer to them. Then we refer to them like picoplankton, nanoplankton, microplankton, and mesoplankton. And mesoplankton is what we consider to be a net zooplankton. What is most commonly is referred as zooplankton. But there are also smaller categories of zooplankton that can occasionally occur in our samples. So sampling with a net of 100 micron, and this is the net we use in the Baltic Sea most commonly if there is no special research idea behind zooplankton sample, and if it's just monitoring or regular screening, then it's 100 meters. So we are cutting it like in between. So some of the rotifers, ciliates, small zooplankton species and juvenile stages are in our samples, and they also contributing to the mesozooplankton sample. So what we have essentially here when sampling with 100 micron net is micro, meso, and macro, if you're lucky, specimens. Macro are those that are like jellyfish, mysids, those that are larger than 2 centimeters. So we have a mix of different size groups in in the samples that we are usually referring to as mesozooplankton sample. But this way of sampling is not equally efficient for all this size range. We say that, okay, if the net is designed not to let through a specimen which is more than 200 micrometers, then the highest efficiency that we have, considering that like they are non-movable, would be for this category. And that's why when analyzing data for zooplankton abundances, this category would be most reliable to base your analysis upon, because efficiency towards smaller animals is lower, they can go through the net, and efficiency towards the larger specimens is also smaller because they are movable, they can swim away from the net. So again, a compromise between the ecology and logistics. And size-based sampling and size-based analysis is kind of common in plankton research, and it goes not just for zooplankton but phytoplankton as well, because the way of collecting samples is tied up to filtration. So it's only water bottles that take all that are in the water column, and then you sediment whatever is there, getting everything from bacteria, eventually, if you are sedimenting it in the right way, to any kind of larger organisms, mesozooplankton organisms. You won't sediment a jellyfish in this way, but theoretically it's possible. So this is the only way to get the total, really total zooplankton or heterotrophic community in your sample, but you will get also a phytoplankton as well. So it will be a total plankton community in this case. All other methods of sampling, namely nets and pumps, are ultimately filtration. And filtration is always done according to size. It's always some mesh size at the end of this process, which makes a cutoff of the size that will end up in the sample. So this is kind of important to remember when you go for data analysis and try to combine data sampled or data that are coming from samples taken in different ways. Currently we run the analysis and sampling of zooplankton, and it goes both for research on mesozooplankton and monitoring according to HELCOM Combined Manual that is currently under revision. And what this revision is taken care of is biomass values. So we found out that we have a full cows with biomass values that are used for calculation of zooplankton biomass. So we have nice and quite reliable abundance data, but when it comes to calculating biomass values, which are, at least in the monitoring, calculated using constant factors. Like we assume that one copy pot weighs, of this species, of this developmental stage, weighs 20 microgram. That's it. So different labs have been using different factors apparently. It's not a catastrophe, but it's something that happens now and then. And we have to harmonize the biomass values for calculations of the zooplankton biomass if we would be able, if we want to be able to actually compare data from different labs. That was a very interesting discovery which prompted this revision. So here is our major tool for zooplankton sampling in the Baltic Sea, VP2 net with a flow meter. Flow meter actually counts amount of water actually going through the net and not going around the net. So it's very crucial to actually pay attention when you use zooplankton data for any kind of analysis, how this have been used in the data acquisition, because some labs or some monitoring teams sample without, and that's okay, as long as they say in the data that the data were collected without a flow meter. It's better not to use a flow meter than use a non-calibrated or flow meter with false readings. So, but using numbers again and compare data from different labs, it's essential to pay attention to existence of the flow meter in the data collection. And the net is pretty long, as you see here. It's from a shape 3.5 meters. It drags from a depth from where you would like to take a sample to the depth until where you would like to have the sample. So it could be a depth resolved sampling, like between 60 and 30 meters, or it could be entire water column from the bottom to the surface. The monitoring data that we have are coming from essentially all parts of the Baltic Sea. And this is only Swedish long-term monitoring data. We also have stations from all countries surrounding Baltic. And this is where the major difficulty comes comparing the data. Here is the Swedish stations, Bosnian Bay, Bosnian Sea. We have also a station here in the Archipelago Sea, but it's Finnish station. It's very interesting to use it when analyzing our data also. We have Baltic proper stations. These are H4 Hemifjärden. It's not technically a monitoring station, but since we have been taking samples within the eutrophication, Hemifjärden eutrophication study, these data are very good and very interesting to combine with the rest of the data. Asker station, inshore station in the northern Baltic proper, and Gotland, and Landsort Deep, the offshore station in the northern Baltic proper. From 2000, yeah, we have also the station in the Kattegat and Skagerrak. From 2007, we have also additional stations in the southern part of the Baltic proper, and it's called Ref V1 for some reason. From all the stations, we have the temperature, salinity, oxygen profile, Secchi depth, chlorophyll, and nutrient data. So this is a standard set of the monitoring data. We also have phytoplankton data sampled on exactly the same occasions. And for some areas, mostly here, we have spurious hydroacoustic data for some years, some stations. From 1999, we have been also collecting an environmental specimen bank, which means that those are samples preserved or frozen, now they are frozen, in a way that is possible to do biochemical or molecular analysis in the samples. Initially, it was for growth assessment, for zooplankton growth assessment, but now they're used for other purposes also, like molecular diet analysis or species diversity in terms of the genotype in the population and so on. So it's functioning as a true environmental specimen bank. Zooplankton diversity. Number of species in the Baltic Sea, as you have heard, is lower than in the marine systems. That's a real main paradox. A recent review covering all zooplankton, including microzooplankton, rotifers, ciliates, and mesozooplankton species, encountered about 1,200 species in the Baltic Sea, which is quite substantial. And it was also a kind of provocative idea that the remain rule does not really work when it comes to the microscopic animals. So it was initially proposed for benthic animals, and it was initially focusing on animals that you can actually count like species like this. Because since in this group ciliates and rotifers contribute most, like 90-something%, it actually shows that the true diversity in terms of this microscopic species could be much, much higher than what's usually appreciated. Among mesozooplankton, we have about 40 species that occur more or less regularly in the open sea. Most of our sampling, again, because of the length of the VP2 net, our sampling sites are restricted to areas with a bottom depth of at least 20 meters. And at least 20 meters in the Baltic Sea is not quite littoral, or it's still coastal zone, but it's pretty deep coastal zone. It's not the same coastal zone as people who are working with microphytes, for example, would refer to. It's not like physically, it's not the same environment. But also what we have here interesting is a gradient, north-south gradient in terms of the species diversity, or species species diversity as a total number of species. Here is the Bosnian Bay, Baltic proper, and Kattegat. So when it comes to the mesozooplankton, they obey the Raymond's rule. They increase towards the marine areas. Proportion-wise, we have very good contribution, or like sizable contribution of crustaceans, cladocerans. Here, the yellow field on the pie chart. We have them almost as much as we have copypods, orange field. Another thing that you can see here is that the blue segment, rotifers, are kind of decreasing towards south. Rotifers are normally limnetic species, so their contribution here and even here are measurable. Contribution in the southern Baltic and in Kattegat are almost non-existent. Again, it's in terms of the number of species. In terms of the biomass, it would be even stronger trend. Another thing that you see here is increase of the red segment towards south, in the southward direction. And red is mostly meroplankton here. Meroplankton, larvae of benthic animals. Benthic animals have complex life cycles. Like 85% of all benthic animals have planktonic larvae, which means that it also reflects the diversity of benthic animals in the south, and as expected. So there is nothing major, like striking uniqueness in this distribution. It's more or less as expected. Protozoans. Now I will go very fast through the groups that we have. And then when I'm done with this, we have a 5-minute break for tea. So, protozoa of estuaries. Ciliates are very, very abundant and very, very diverse in estuaries, including Baltic Sea. Though those are complex single-celled organisms, they could be free-living like Paramecium. They could be also sitting like Vorticella or Stentor. Solitary and colonial, they can build colonies like Didinium, for example. Most of ciliates are soft, have no hard parts. They graze, they're very important bacterivores. They graze on bacteria and they provide, they are themselves food for mesozooplankton. So they can really consume bacteria very efficiently and very efficiently with a very high energy transfer efficiency from bacterial biomass to ciliate biomass. So they are essentially predators of the microbial food web. The group in the Bosnian, in the UMEF, UME Marine Research Center, works quite much on the ciliate bacteria interactions. Rotifers, those are smallest multicellular animal on Earth, which is kind of funny, I think, because the body size is really crucial in terms of the feeding, in terms of the food web interactions. They also could be solitary and colonial. Here is the Caratella and Syncheta, two most commonly studied or most commonly referred to group of ciliates in the Baltic Sea. This one has a lorica, and this one is soft-bodied, which is loricated and alloricated forms. They also can graze on bacteria, but less efficient than ciliates. They, although, can graze on ciliate quite nicely and on small zooplankton, or on small phytoplankton too. Occasionally they may dominate. We have two rotifer blooms in the Baltic, one in the beginning of summer, end of the spring, and another is in the beginning of fall, when they both blooms are dominated actually by different species. This is a spring bloom and this is an autumn bloom. Mesozooplankton, those are most prominent and those are most studied and those are most referred to as zooplankton. Copypods, cladocerans, evadna, ostracods, they could be present in plankton samples, zooplankton samples as well, although not that frequently. They are more common in Bosnian Bay and in the southern Baltic than in the Baltic proper. Interesting is also it's two different species in the Bosnian Bay and southern Baltic. Mysids, those also are crustaceans and feeding on phytoplankton as well as zooplankton. They're also very good food for fish, as we know. Copypods, which is usually referred to as a, okay, here we have copypods, but copypods have very complex life cycle. It's one of the most complicated life cycles among the invertebrates in general. So they have 13 life stages, and one metamorphosis. So from the eggs, they go through 6 nauplial stages. Then metamorphosis occurs here with a total body restructuring, total shift in terms of the gut structure, a total shift in terms of the swimming behavior and other things. So this is metamorphosis that essentially comparable to hatching from the egg. Some nauplial stages up to the 2nd or 3rd or sometimes 4th are non-feeding stages. They don't eat at all. They don't have the normally developed alimentary canal, and they cannot process food. What they do is they use yolk from the egg that they received from their mothers and develop through several molts until they reach the feeding stage. Linking nauplial abundance of these stages to any kind of feeding conditions is senseless, simply. Then copypodite stages. Those resemble the adult already a little, although they have less segments, they have smaller body size, they have less developed feeding apparatus, they swim not that good. But one way or the other, they develop into the adults with the sexual characters apparent somewhere here, depending on the species. And then everything starts again. So some copypods in the Baltic make this cycle only once a year, the univoltine populations, like Pseudocalenus, for example. Other copypods can have 4, 5, 6 generations a year, like every Temera. Those are two extremes, more or less. Other species somewhere in between. So And this goes for calanoid, cyclopoid, and harpacticoid copypods. Those are subclasses of the copypod class. Harpacticoids, mostly benthic animals, but they're also planktonic forms, and they're also planktonic species of harpacticoids that could be found even in a Lancet deep area. So pretty far from the bottom. Cyclopoid are interesting predatory copypods, whereas most of those copypods are considered as herbivorous, although they can feed on ciliates and heterotrophic nanoflagellates and other heterotrophic component of the plankton. They are mostly considered as herbivorous. Cyclopoid copypods are true predators. They feed on nuclei of the same species or other species. They feed on small cladocerans. They can feed on small rotifers. They can feed on anything that is actually animal food. Those are very common in coastal areas, much more coastal that we sample as coastal. They are also common in the Bosnian Bay and freshened areas, like in Hemifjorden. We have in stations that are close to the Malaren entrance. We have quite many cyclopoids in spring, for example. But they are associated more or less with fresh waters. The only cyclopoid copypod that just came up to my mind is, which is truly marine, we have in the Baltic, is Oethona. Oethona is not predator. It's mostly, it's very like actually microphagous species feeding on small algae. It can feed on ciliates as any other herbivorous copypods, but it's exception from other cyclopoid copypods that we have in the Baltic. Right. And where I was when talking about the nuclei, as in any age-resolved like demographic structure in a population, copypods have a lot of juveniles, and then a biomass or number of both biomass and number actually of each next life stage is decreasing. If we say that, okay, we have that much of copypods in this area, it means that most of the copypods that we include in this number, in this biomass or abundance number, are coming from juvenile stages. What is not the case that most experiments are done with adult copypods, again for our convenience. Most of samples that we take from the water for different kind of analysis, like stable isotope analysis or biochemical analysis, we also try to pick those that we can distinguish in terms of the species and we can distinguish in terms of the life stage. We pick the biggest ones. But what makes it not actually quite representative for the community is that the proportion of this in the population is the lowest. So whatever we measure for a least representative part of the population. Yeah, some other copypods in the Baltic Sea, just overview. Pseudocalanus acuspis, which has been referred to as elongatus for 120 years. Now we have analyzed all the pseudocalanus in the Baltic Sea, all the pseudonec stations and samples that we could find. Tove Holmborn actually wrote this paper as a part of her, not as a part of her thesis, it came afterwards actually. So pseudocalanus samples from everywhere in the Gulf of Riga, Gulf of Finland, Bosnian Bay, south, east, west, and they turned out to be just one species, Pseudocalanus acuspis, which also has extremely low diversity in terms of the genetic diversity, in terms of the mitochondrial cytochrome oxidase, the barcoding gene. Which means that species which has been referred to as Pseudocalanus elongatus and compared to this Pseudocalanus elongatus in the North Atlantic, in the Pacific, in the, you name it, has been all the time totally different species. I think it's very fascinating story, actually. Limnocalanus, this is the copypot of the Bosnian Sea and Bosnian Bay. Fish there feeds on Limnocalanus. As salinity decreases, actually fish feeding condition increases because Limnocalanus is freshwater of freshwater origin originally, and Limnocalanus is actually thriving in low salinity conditions. This is also kind of in contradiction to what usually linked to fish feeding conditions in the Baltic. Low salinity, poor fish feeding conditions. Not always. There are areas where low salinity actually improves fish feeding conditions. Acartia longiremis and Acartia bifillosa, we still refer to them as Acartia longiremis and Acartia bifillosa, although it's a complex of different species. Every Temera finis is a nightmare in terms of the taxonomy because it's apparently not just one species, but definitely two. And they have such huge genetic diversity in the Baltic Sea, so that the diversity within the Baltic is comparable to the diversity between the Baltic and Atlantic east coast in the States, or Atlantic coast along the Europe. So this is, think of the implications. So if we say that this species is increasing or decreasing, it actually means that it's a complex of species that are increasing or decreasing, not necessarily one species. And complex of species can increase or decrease because one particular species starts to take over in this community. So it's not, it doesn't make it difficult, the diversity, both species and genetic diversity. It doesn't make it easy to actually interpret the long-term variations. Yeah, southern Baltic copypods. Paracalanus, Mitridia, Lucens, Centropages, Temora, those are usual, and Itona that I said, it's a cyclopoid copypod in the southern Baltic Sea. So those are copypods contributing most to the southern communities and Kattegat-Skagerrak community, Bornholm Basin community. So those are not the same as in the northern Baltic Sea, which also makes it difficult to actually compare the copypod abundance, saying that it's okay, it's just copypod abundance. It's different species with different adaptation capacity, different optima, temperature, salinity, and so on. Baltic cladocerans, Bosmina maritima, which is actually quite interesting species, understudied to my, in my opinion, and it's truly endemic species in the Baltic Sea. It's Chidariids, coastal samples. Cercopages, very predatory and very difficult, very differently looking species, feeds on other zooplankton, and in the freshened area, it's Daphniids, different species. And those are mostly in the more or less fresh areas. Baltic proper and southern areas, we have mostly podonids of different kind, poor taxonomic resolution in all zooplankton data, doesn't make much sense in most cases, and Penilea, which is invasive cladoceran in the southern Baltic, Kattegat, not here. Totally ignored part of the Baltic plankton, larvaceans. Nobody cares. I actually don't get why, because if we rank species, different species from southern Baltic, it's Gdańsk Bay, and eastern Baltic Sea, close to Gulf of Riga entrance, I think. So if we rank the species according to their biomass, larvaceans, Fritillaria borealis, actually comes up pretty high in the first 6 species. Different from all other mesozooplankton species, they feed on bacteria directly. So those can actually utilize bacterial biomass, surpassing the classical food web, and transfer directly this bacterial biomass to the fish. That's very, very unique for the Baltic Sea. The only other species that can do it are comb jellies. Those small comb jellies that we have in the Baltic Sea, they have a similar feeding behavior. Meroplankton is also understudied part of the Baltic zooplankton. So we have in some periods when all the barnacle, for example, reproduce, and they do it quite synchronously. It's a pulse of huge biomass coming up in a water column, spreading around with water currents, and used. They're of the size of a small coffee pot. They're perfect food for fish. As you see, there is a little bit of green here, meaning that it's been eaten, and it's been eating something green, which is probably algae. So they are both grazers and fish food. Those pulses are quite predictable. So they are coming, for example, in our northern Baltic proper samples, they are coming in May, June. 2 weeks, very strict window, and this is also the time when fish larvae hatch. So why not? Other larvae of invasive species are, other larvae of benthic species are also kind of ignored in all the analysis, although this could be also a perfect way of actually detecting invasive species, benthic species in the ecosystem. Because here you have an integrated sample of everything from a large area. They are mixed up, they are taken from different bottoms. It can take a number of years before you will be able to actually find this specimen of a crustacean or a mollusk on the bottom. But since they are producing tons of larvae, thousands of larvae, each individual, it's much more probability that you can detect the larval stage than you can detect the adult stage. So when talking about this spatial dynamics, it's usually horizontal and vertical distribution that are addressed. Horizontal distribution, migration, horizontal migration in terms of the inshore, offshore migration, spring, summer migration, summer, fall migrations. This goes mostly for inshore zooplankton, the zooplankton that actually moves from the shore towards the open sea station, open sea. Vertical distribution, also a part of the spatial variation. They are not uniformly distributed along the water column, as you guessed. There are specific depths where we have more and other depths where we have less. Seasonal population development is also a part of the distribution in terms of the seasonal dynamics. And here we have an overview of the zooplankton stocks and the Baltic Sea. The stations on the x-axis are ranked from north: Bosnian Bay, Bosnian Sea, Åland, Gulf of Finland, Himifjärden, Askö, Landsort, Gulf of Riga, Eastern Gotland Basin, Litauen, inshore and offshore station, and Bornholm. So it's more or less north-south gradient. Abundance and biomass. So summer period for everything, and the whole data set is based on the long-term, summer period, June, September. I'm standing here. And the long-term data set means that this is as long as data go back in time. For some stations like Askö, it's 76. For others like Litauen, it's 2000. But basically, it's all available data sets. So as you see, we have relatively low abundance in biomass in the north, increasing towards the Baltic proper. And in the Baltic proper, they are the highest. Towards the Bornholm Basin, they're decreasing, and they have been relatively low here for a number of years, so it's not a recent phenomenon. Another thing that you can see here is a more or less horizontal gradient from the western Baltic to the eastern Baltic. Hemifjorden, for example, represents the westernmost station in the Baltic Sea, and Gulf of Riga represents easternmost station in the Baltic Sea. So here we also have kind of parabolic distribution with lower, generally lower numbers and less variable numbers in the middle part compared to the coastal areas. This was quite interesting exercise also to look how variable altogether the zooplankton biomasses are. In terms of the seasonal dynamics, mesozooplankton community structure and spatial and seasonal variations. Here we have data for our Swedish stations. Units are biomass. Scale is identical in all graphs. This is some specific year. I don't remember which one. I think it's 2007 or 2008. It's not long-term data. It's for a specific year. But they reflect more or less well the increase in biomass values in summer period, being highest in July, August, almost everywhere. July, August are the highest. Here at Asko, actually, the highest period is beginning of summer compared to the second part of the summer. Lancet data also peak in the beginning of summer rather than second part of the summer. What actually shows here also relatively lower values for Bornholm station. It's for BY5, which is here. Relatively low values compared to the values in the middle part of the Baltic. Yeah.
If there is any biomass, that means probably there isn't any
no sampling.
Yeah.
So this is actually what complicates this long-term data analysis, that the samples are not taken at the same with the same frequency by different labs. So for Germany, for example, no samples in August ever.
But they're all taken with a WP2.
They're taken with the same net. They're taken from the same depth horizons. They are analyzed using the same counting methods and same biomass conversion factors. So those are the data are standardized in terms of the sampling gears and analysis, but they're not standardized in terms of the frequency. And for some data, for example, here we have the error bars, meaning that we take samples twice a month. But this is basically, also here they have, in Umeå they also take samples with some frequency in summertime that is more frequent than once a month. Yeah.
Does it also matter when during the day the samples are taken, since they
in the guidelines it says that samples have to be taken during the light hours. Light hours also kind of variable, but usually due to logistics, ship goes to the station, samples are taken around lunchtime, and ship comes back. It's almost the same logistics for us for Lancet tip. When we arrive at the station about noon, take samples, and so it's. I wouldn't be worrying about the time of day in terms of the data comparability.
But this is specific year. It's not long-term mean.
This is specific year. Yeah, it's not long-term data. What we see also here is the relative contribution of different groups like copypods, cladocerans, rotifers, and others in the same way as we saw for the species diversity. Here we have a huge pulse of cladocerans that essentially contributes quite a lot to the total biomass in terms of the year estimate. Cladocerans are also quite common here, together with rotifers. Here, copypods become more dominant. Here we have mostly copypods, very little cladocerans, but spring bloom of rotifers that contributes to biomass quite a bit, and also fall bloom of rotifers. The same in the Lancert deep. So those are two stations, and those are two figures for this. The southern station, it's not a good station in terms of the monitoring. I think it wasn't the right choice for the station because it's very shallow. It's on only 70 meters depth. The stuffs that we get in those samples are more representative for benthic than for planktonic system. BY15, for example, yeah, right. This is BY15 for comparison also. Mostly copypods, but also a huge Bosmina peak in summer. It's the August peak of Bosmina. Bornholm, still a contribution of cladocerans in the beginning of summer, but the rest of it is mostly copypods. And the Anholt Kattegat is essentially just copypods with some meroplanktonic larvae contributing. So to the seasonal abundance, different groups contribute differently depending on where we are in the Baltic Sea. Vertical distribution of zooplankton. If you look at the vertical profile for abundance or biomass, we see that, for example, daytime and nighttime distributions differ. So the yellow is day, the black is night. During daytime, most of the biomass is concentrated in a pretty thin and dense layer around the thermocline. So for this particular sampling, the thermocline was around 16, 15 meters. So they're just sitting on top of the thermocline because the temperature is still comfortable. They don't want to go below the thermocline. And the fish predation is minimal. During nighttime, some of them migrate to the surface, some of them don't actually, or they can migrate but from the deeper layers. When we look closer into what groups migrate and how, we see that copypods, this is who contributes to the 15-meter thermocline peak. During daytime, they're sitting there. During nighttime, they are distributed in a trophogenic layer, more or less. Cladocerans also migrate, although to a smaller extent, but they migrate also closer to the surface compared to the copypods. Rotifers are almost non-migrating, so there is no essentially difference in the vertical distribution between day and night. Now we look at what copypods actually contribute to those peaks. Ignore cladocerans and rotifers, look at copypods only. Yeah. And copypods and this station was a little bit south of Gotland, in the Gotland basin anyway. So those are the copypod species that we had at the time of sampling, and day and night distribution. 5 species: Centropagus, Acacia, Erythemura, Pseudocalinus, and Temura. So among all the species, the Erythemura, the green one, night and day, actually shows the most differences between the night distribution and day distribution. Acartia is thermocline oriented species. It has a broader range during night time, consistent with migration, but basically this is what sits around 15 meters in the thermocline. So there are species-specific differences, group-specific differences in vertical distribution, but there are also demographic differences in the vertical distribution. So here are the different life stages of copypod, or different ages of copypod. Younger stages appear to migrate the most. So here they are ranked from eggs. Eggs should be actually on top here, and then C1 and so on. Oh no, eggs here indicate the egg production by females, which occur along the sampling profile. So what we have is that the stages that are youngest, blue and green, they differ the most between the night and day distribution. So species specific, age specific, group specific differences in vertical distribution. So depending on where you go with your sampling gear to get a zooplankton sample, depending from which depths you will take it, from here to here, or with water bottle just here or just here, you will get very different communities in your. Yeah, based on the vertical zonation of mesozooplankton that we have in the Baltic, they could be classified in major ecological groups like upper epipelagic, lower epipelagic, and species with a deep and broad distribution, meaning that depending on stage they can be wherever in the water column. For daytime distributions, the maximal depth of maximal abundance, which is a depth where you can find the most of the population biomass concentrated. For Pseudocalanus is the deepest, and then it goes according to lower and upper epipelagic. And during night they change, but also they change quite a bit here, but they don't change much here. It's also reflected in variability. Seasonal dynamics. In spring, following the phytoplankton bloom, and phytoplankton bloom here is shown as it's not shown here, but okay, we have a temperature production of phytoplankton instead. No, it's not phytoplankton, it's ciliate production. We have temperature as a proxy for seasonal, for changes in physical conditions during the season. First, we have a bloom of ciliates in spring that follows phytoplankton spring bloom pretty close. And this spring bloom of ciliates is actually the first zooplankton biomass in the water. So they enjoy unlimited food conditions. Due to low temperature during this period, zooplankton biomass is starting to grow relatively slow. So during springtime, the zooplankton growth is mostly limited by temperature conditions. But as temperature goes up and egg production goes up, again, females that are there to produce eggs, they don't have any food limitation during this time. Zooplankton starts to increase. By this time, ciliates decrease, partly due to the predation from zooplankton, and most of the zooplankton production comes as expected during summertime. Most of the zooplankton production, and those data are for Lancert deep, so it's open sea Baltic proper. Most of the zooplankton production during springtime, May, June, or beginning of summer, is coming from rotifers. Rest of the summer, it's copypods, they contribute most. What is interesting here is that those y-axes are almost the same, so 25 here and 20 here. Ciliate biomass is comparable to that of mesozooplankton biomass, which means that the grazing on phytoplankton is mostly from ciliate. Ciliate production transferred to zooplankton production, and again, the ciliate production is comparable to zooplankton production. It's actually higher, if strictly speaking, it's higher. Temporal trends. What we have here, yeah, I will go into how the long-term changes in zooplankton look like, starting from the up north Baltic Bay and going down to the Baltic proper, Central Baltic proper. So here we have biomass change in total zooplankton biomass change in copypods. Looking at trend, it's not much here. Looking at trend on copypod biomass, the increased copypod biomass was observed in the last about 10-15 years. Bosnian Sea, now we are there, a little bit south. The total biomass trend looks somewhat similar to what we have here. But by contrast, the decrease during the last 10 years is significant. What is even more interesting here is that the biomass that we have here is composed by mostly both minor, and biomass that was here was composed mostly by copypods. It's easy to see if we look at specific biomasses by cladocerans, red, and copypods, blue. So as one increases starting from somewhere in 2000, the other one decreases. Even easy, we can calculate a ratio between the two and see that this decrease is actually quite dramatic. Northern Baltic proper, coastal station. This is the Askio station. This is the best data set ever in the Baltic Sea in terms of the length and sampling frequency. So this we should be proud of, really, because no one else takes samples every second week. Total biomass is actually monotonously decreasing from middle 80s, and the contribution of copypods to this biomass, while increasing somewhat up to 90s, starts to decrease in like for 20 years back. But the overall decrease in trend for the copypods is also significant here. If we kind of think in numbers here, then from about 90% of copypod biomass contributing in 80s here, now we have only 50% of copypod biomass contributing to total zooplankton now, or in the last basically 5-10 years. So this is a difference, 90% of the community or 50% of the community. And this decrease in terms of the copypods was accompanied by increase in not just cladocerans, not that much actually of cladocerans, but mostly of rotifers. So those are quite interesting data, I think. So it's extremely sharp shift in beginning of 90s towards a totally different state. So now rotifers, and here on the y-axis we have B1 red dots, on the right axis we have Himifjärden station, blue dots, but they all are essentially the same, so they all show the same pattern. What's happening now is that it goes back, both stations. So the rotifer abundance today is back. There are a few years missing here that were not in the graph when I was making it, but now we know that we are back to the same level. So two alternative states. The interesting part is that Syncheta also behaves in the same way, but only on one station. It doesn't behave the same way on H4 station. It doesn't show any trend at all on H4 station. And Syncheta is more marine species. So the most tempting is to actually relate this to salinity fluctuations, but it does not correlate with salinity, at least not in this, not straight way. This increase could be also associated with eutrophication. Of course, rotifers are indicator species for eutrophication, but it's still not quite clear what we are referring to as rotifers, because generally those are the most known group for having cryptic species. What we refer to as Keratella cochlearis is in fact like 25 species. It's not about 2 species or 3 species. We don't know how many they are. And they are different, those the individuals that are identified as cochlearis or Keratella. They are different at the level, or genetic differences are at the level of species differences. So it's less than 95% similarity in ribosomal genes. So, Lancer deep. Lancer deep data are more problematic because they are essentially composed of 2 different data sets. One data set that we have is coming from sampling that was conducted in beginning of summer, and the second data set is coming from sampling that was conducted in averaged over the whole summer. If you consider only May-June data for to make it comparable, it will be the same essentially. The error bars will be slightly low. The totals here would be probably slightly low. I don't remember. I did this, but I wasn't able to find this graph. So in theory, if we have less frequent sample here and more frequent sampling here, we can sacrifice some of the data from this data set and see if they are follow the same pattern. But they do follow the same pattern. So what we have on a Lancer deep resembles what we have essentially in the Bosnian Sea. Increased about the same time also. Copypot biomass followed by decrease in copypot biomass and compensatory increase of cladocerans. So the community, again, while it was formed by copypot biomass before 90s, it's formed by cladoceran biomass to large extent now. Again, the interesting difference in biomass values, if it was like about 20 in the beginning of 90s, now it's just 1/5 of what it used to be in the 90s. So the absolute numbers are actually quite dramatic. That was about the long-term data. Questions? Crystal clear. So here is basically approaches listed what have been used to study feeding of zooplankton in the Baltic Sea. And on the first place is, of course, experiments. Most of what we know about feeding and food interactions or trophic interactions in the Baltic Sea involving zooplankton are coming from the experiments. And most of the data that are used in the models are also from the experiments. Representativeness questionable again, because what is selected for the specimens that are selected for experiments are not necessarily the most representative specimens that are of interest if you estimate flows between the different compartments in the system. Next most popular method, I was trying to kind of put them in an order of popularity or commonness. It's very subjective, of course. Next is stable isotopes. Good because they are about the in situ conditions. Bad because the trophic level estimate does not imply the diet. Migrations can obscure the stable isotope data in terms of the explanatory power to actually infer diets from stable isotope analysis. But again, the good sense that this is about in situ conditions. Next is gut fluorescence. Nowadays, nobody almost uses it. Before, it was quite popular method to measure actually pick a plankton, stop put it in a fluorometer and measure how much chlorophyll is there. Allows to estimate herbivory, poor in terms of the corrections for chlorophyll digestion, but still good because it's for in situ conditions. Fatty acid analysis, amino acid analysis also here. As far as I know, amino acid analysis has not been used for in the Baltic, but fatty acids were used. Diet of herbivores, indirectly, fatty acids that are channeled to the consumers without transformation could be used indicative, could be like indicative of the algal food sources or groups of algae. Good because it's for in situ. Less good because it's hardly calibrated using the species and species of both consumers and food that are relevant for the Baltic. Molecular diet analysis, very recent, when we go and pick a consumer and see what kind of DNA it has in the stomach. In a way, it has same limitations as gut fluorescence analysis because some of the DNA get digested, disappears, difficult to actually quantify amount of food in the stomach or calculate the consumption rate from the amount of food in the stomach, amount of DNA in the stomach. It's more stable than chlorophyll, so the issue of degradation is less important, but the rest of it is still there. Developing, someday it will be standard. Yeah. Feeding modes of zooplankton that is relevant. Bacteriivores, as I mentioned, appendicularias and larvaceans. Bacteria nanoparticles in the water, so this is virtually unknown pathway in terms of the food web. Detritivores, those that are feeding mostly on organics, dead organics, dead bodies. Meroplankton larvae. But not exclusively, like polychaete larvae are very good in it, barnacle larvae are less good in it. Herbivores, most of the collonoid copypods, cladocerans and rotifers are in this group. Carnivorous, cercopodes, shitagnats in the southern Baltic, and most, of course, those are strict. These are relatively strict group. The rest are jumping from one food source to another, and collectively I should be called as omnivorous, all of them, essentially. Stable isotope analysis is good for some communities, but not for others. Like, for example, here we have mapped carbon 13, nitrogen 15 signature in a microplankton food web. The two, or not microplankton, but just a pelagic food web, planktonic food web. The two consumers here is cercopodes, which is used as a pure predatory group. It feeds on other zooplankton, preferably crustacean zooplankton. And the whole question here was to find out what eats comb jellies, which are here. So classically, comb jellies are the plantivorous, and the question was if they compete with cercopodes or other zooplanktivora for herbivorous zooplankton, or if they don't. Apparently, they cannot compete, and under any circumstances, they cannot compete because they are feeding on small particles. Most of them are smaller than 20 and 6 micrometers, which are here. But this is for summer community. The same groups and same system for winter communities is less convincing, because here we have also appendicularians as consumers here, and they're feeding as they should be feeding on stuff that are less than 6 micrometers, of course. 6 micrometers. But the rest of it is just one big lump. So to go there and actually find out what's happening there, one needs compound-specific analysis of stable isotopes, which I'm not going to talk about now. The relationship between phytoplankton and zooplankton in general. I don't remember how long back this data go, but those are two stations that I compared. One is Askøy, another one is Himifjorden. If we compare zooplankton stocks and phytoplankton stocks from different stations and regress it against the phytoplankton stocks, then for each particular station, whereas for each particular station we don't have practically any relationship, at least during summer. But if we set up several stations, so then some relationship can arise, but it's very fuzzy, and I don't actually know how informative this relationship. Basically, what this figure shows is 2 levels of primary productivity in 2 areas, and zooplankton is actually adjusted to the areas of primary productivity. So between area comparison is possible, but not the within area. Other types of zooplankton interactions, if you look at not just total phytoplankton, zooplankton, but species-wise, different phytoplankton groups, different zooplankton groups, then we can see, for example, that some positive linkages between the erythema and cryptophyceans, praesiophyceans could be found. Cladocerans are mostly related to temperature. They basically eat whatever is in small class categories of phytoplankton. Acarcia are more related to chrysophyceans, and Eglyphnophyces, which is a very large phytoplankton cells. Acarcia can take it, that's for sure. So rotifers, similar to cladocerans, do not seem to be related to in any specific group, but rather to, again, they're not selective towards a group, but they're selective towards size, prefer less than 10, probably 7 micrometers. So abundance comparisons do not make much sense when you want to find out what's happening to the population. And why they don't make much sense? Because what we measure as a population size, and this is our abundance or biomass values, is affected by reproduction from one hand, as you remember from the ecology course, and mortality, natural or accidental on the other hand. So it's a flow, and because of this you can have enormous rate of flow through the system, enormous rate that could support any kind of upper consumers, enormous rate that can take care of any kind of algal bloom, but it won't be showing in the population size if mortality or fecundity are affected not in the same way. And that's why what we have in our monitoring samples for biochemical analysis, I think it's invaluable, and it becomes more valuable with each year. So first was Zurich who started to collect samples. They were in the jars for urine samples. I think they're still there. Yeah. They're very different from all the others. Yeah. And to look at the production, we can look at growth rate, because growth rate is here, is not affected by predation. It's only the population size that is affected by predation. If we can measure growth rate of each particular component in our food web, we can actually tell much more about the actual energy flow between these components. Okay, and here what we have for Nauplea. Nauplea growth could be measured by using the biochemical markers. In this case, it's RNA-DNA ratio marker. And what we have is Nauplea for 2 species. We have different parameters. As a dependent variable, we use the growth index. As independent parameters, we use climatic indices like now and Baltic Sea index, temperature, salinity, and a list of phytoplankton groups to see what's happening there. Essentially, what's happening there is that, surprisingly, and this is where we first started to think of cyanobacteria as a source, not necessarily as a threat to zooplankton production. Surprisingly, abundance of filamentous cyanobacteria had positive effects in both cases on Naupleal growth. It was very difficult to explain why. First, because they cannot feed on cyanobacteria for sure. What they do, Nauplea of this age, and those were feeding Nauplea, they feed on bacteria. They cannot take filaments. But what can happen in a system is that filaments take up extra nitrogen. They get rid of this extra nitrogen by releasing it in the form of either dissolved organic nitrogen or dissolved organic carbon. And this fuels the bacterial and picoplankton production in the system. And this picoplankton production actually is becoming available for the smallest components of zooplankton, larval, Naupleal stages, rotifers, and to some extent cladocerans. The rest of it can go also not just to picoplankton, but also to normal phytoplankton and support, in a way, production in the zooplankton communities. It can also go through ciliates and again support zooplankton communities. It can go all the way through the bottom and support even benthic feeders like Monoporea. So this is how we can explain the positive linkages between the cyanobacteria blooms, which is known as poor food, again from experimental studies, low in sterols, and crustaceans are very dependable on sterols, low digestibility, low manageability, so it's difficult to grab and eat. So eventually all this should, in theory, support the fish production. And Nisha, who is on maternity leave now, actually has done a very neat study measuring, using molecular diet analysis, the picocyanobacteria in copypot Nauplea. So what she found is that they really feed on it and they eat it very efficiently. Now I will make a case of positive effect of filamentous cyanobacteria blooms on zooplankton stocks, not the stocks, on zooplankton body composition. Transfer of biomass from cyanobacteria to copypots in this case. So here what we have for cyanobacteria blooms in Asker, similar also for Lancer deep, but in this case both copypot samples and cyanobacteria samples are coming from the same station. So what we have is, starting from the beginning of 90s, end of 80s, we have a very heavy increase in all cyanobacteria species that are dominant cyanobacteria in the Baltic, which are Aphanisomena, Nodularia, and Anabina. At the same time, we have stable isotope nitrogen 15 signature in copypots decreasing like this. And red dots here are not monitoring samples, but samples coming from independent research campaigns, when people just go there, sample zooplankton, analyze them fresh. These black dots were analyzed after many years in formalin storage. So there could be potential effect on the isotope signatures, storage effect on isotope signatures. So just for comparison, to see if we're on the right track here, we'll plotted all these independent studies. There could be more, and I will try to locate them also. And of course, the correlation between the increase in cyanobacteria and decrease in nitrogen, light nitrogen in copepods have pretty decent for bioecological standards, with like R square 0.7, and actually no indication of non-linearity. So what we have is a nitrogen fixing cyanobacteria supports zooplankton growth. I'm not saying that they are good for zooplankton, but given the rest of the phytoplankton community, they have rather positive effect in terms of affecting other phytoplanktons, in terms of transferring nitrogen from pumping nitrogen in a rather nitrogen limited system. The net effect is rather positive than negative, and given the other circumstances in terms of the salinity fluctuations, in terms of the eutrophication effects rather than effects on sands. And since zooplankton is a base for fish feeding, so growth rate of fish related to the growth rate in fish. So we have a pretty high period again, which corresponded to high proportion of copepods in zooplankton stock. And we have low period, which corresponded to, oh, the other way around. This is a high proportion of copepods in terms of the pseudocolonies in zooplankton stock, and we have the low period, which corresponded to less pseudocolonies and more acarta. But those are data for the Gulf entrance of the Gulf of Finland station L7 there, where pseudocolonies is really a significant component. If we'll do it for example, Asker station, pseudocolonies is not going to be a significant component there. It's not going to explain much because it's too shallow a station for pseudocolonies to build a large population there. Yeah, and from the same paper, zooplankton fish linkages, they found, not surprisingly, that pseudocolonies is associated with good herring conditions, and erythemora and acartia are associated with more mispracticing and wet herring conditions for the herrings over there. Again, depending on what community you are looking at, different zooplankton species are going to have positive or negative effects on the same fish species throughout the Baltic Sea. Invasive species, very short, very one slide. Now, like 15 years back, we had just cercopods. Now we have two other invasive zooplanktons that are coming more and more often. Actually, Vadne now, in August, is almost dominating species. And these zooplanktons essentially share the same trophic niche as cercopods. They are predators. By contrast to cercopods, which is relatively large cladocerans and can feed on copepods, those two are feeding on nauplial, rotifers, small other cladocerans like Bosmina and quite small stuff. But they're still sitting in the same place in the food web in the Baltic Sea, which is kind of simple. Here is a phytoplankton, zooplankton, young fish, adult fish. Plenty of effects. And effects could be both like down to the bottom and up to the top. For cercopods, we found that young fish is not suffering when it's controlled. When cercopods is controlled by large fish, which means that large fish stocks should be relatively should be substantial to actually make this control. A little excursion in Swedish zooplankton research. It started actually a few years back with my Finnish colleague. We started to count who published more Finns or Swedes about zooplankton. Normalization was needed because generally publication rate is going up regardless of subject. So whatever was published about the Baltic from the Swedish side is this black line. Whatever is published about the zooplankton in the Baltic from Swedish side is a blue line. So the red line is percentage of the blue into the black. So this increase is significant. I don't know who did that. Huge contribution, but it was there. I forgot. I knew, but I forgot. So, Swedish zooplankton research is on the rise, even considering the, yeah, and Finnish goes down. That was like the most of the results that we got during this day.
Waste money.
What?
Why waste money if somebody else is doing it?
Compensatory effect, truly. And collaborations actually would contribute equally. So it cannot be explained by collaboration in any way. Yeah. So, interesting phenomenon. I don't know, but Finnish research in mid-90s was really, really strong. I would...
Blood group.
Yeah. Blood group. Exactly.
Juha Flinkman.
Yeah, Juha Flinkman and girls. The girls. Well, and then I started to kind of look, okay, here we have increased number of publications. Do they deal with exactly the same things, or they kind of look also around for some unknown existing parts? So first papers, so the problem is that the Web of Science goes only to 70s properly. The law is not interpreted. So first papers that were highly cited, and there, it's not a great literature again. In the 70s, it was still a lot of stuff published in reports. First papers deal with sampling, sample preparation, counting techniques. Essentially, we are still using them in a pretty much the same way. Individual made biomass estimates, individual weight estimates. It was extremely popular topic for like 5-7 years in Finland, which coincided with Kalevi Salonen made a beautiful machine where you can measure carbon content of a single rotifer. You just pick this rotifer with a loop, put it in a chamber, it burned up, and you have a carbon content of a single individual. It's still a very unique machine, like worldwide. Then distribution, of course, population dynamic production. When they developed all the sampling techniques and so on, so it was a very long period also during the 70s, 80s, 90s, when people were just collecting descriptive data. Now it went up, now it went down. Here it's that much, here it's that much. And of course the species diversity, morphological, taxonomical studies, ecology. Invasive species actually came quite nicely into this part in late 90s. And then invasive species also kind of filled the trophic interactions field. But it's not like just invasive species. People were trying to actually infer who eats whom. And this was a very good period of mesocosm experiments, plenty, microcosm experiments, feeding experiments. So this is where the feeding interaction study began properly. Food web theories, stoichiometry also came in very handy here about this time. Energy flows models, first uses of stable isotope analysis by Hansen et al. I think it was one of the first uses of stabilizer toxin in the Baltic. Yeah. Stoichiometry, nutrient recycling. So what comes off out of this trophic interaction is the waste that zooplankton produces, how it supports the primary production, and so on. What is the nutrient requirements of different algae? So it gets more and more chemical in the late 90s, and I would say now. So biochemistry of growth, regulating factor of growth. So it's very unusual to find now a paper that describes the population dynamics in some areas. It won't be a story. A story would be if you have added something about why we see what we see there, why it goes up, why it goes down, what's happening up in a food web or down in a food web from these parts. And then genetics and molecular ecology that explains the mechanisms of the phenomenon that they observe. So basically what does the chronology tells you is that the descriptive period is over, measurements, more than just counting samples. Although it's still admirable how efficient some studies were in terms of, for example, the individual biomass assessment for 30, 35 years back. Research becomes more synthetic, so it's not only one group that you, but this goes for any ecological research, so it's not just one group that you look at. Looking at zooplankton, you have to look at nutrients, you have to look at phytoplankton, you have to actually come up with some explanations for the phenomenon observed. But what we have actually holds back is the techniques for sampling and biomass assessment. And those are exactly as they were for 120 years back. So our nets are essentially the same as what they've been using in the beginning of 19th century for krill. It's different mesh size, right? But all the rest is pretty much the same. So the technical part of it is not improved. For phytoplankton assessment, there have been some very good tools like Cytoboy, FlowCam, when you can actually go out in the sea and measure the both abundance and species composition with an instrument. For zooplankton, we have similar, or at least instruments with similar ideology, similar concept, but they're not that efficient and they're not easy to calibrate for specific communities. They also are for more or less large zooplanters, like Calenus. For Calenus, it's perfect. For Calenus, you can really use Zooscan or something to analyze your samples, or plankton recorder to measure the abundances. But for small mesozooplankton specimens or species as we have in the Baltic, it's really challenging. And people hear that, okay, the average size of our puppy pot is 0.5 millimeters. People who develop these techniques, they get discouraged. I think that's it. Yeah, and again, I was kind of thinking, okay, what would be the hot topics for zooplankton research, or very influential topic, not a hot topic, but influential topics. If we can, of course, predict distribution and abundance, that would be great. To predict it, we need to actually understand mechanisms of the environmental factor effects on whatever species we are looking at. We can forecast changes in communities. We can measure out adaptation capacities to actually predict distribution, not only in spatial, but at the same time. Quantifying trophic linkages in situ, it's still a very nice challenge, because what we lack is basic information on who is who. Genetic and genotype-specific functional diversity and diversification mechanisms. What the hell that means this rotifer and everything, or diversity in terms of the long-term fluctuations? Is it the same species clone, subspecies that takes over, or all species just at once thrive and contribute equally to this increased communities? We don't know. And what does this diversification means for future communities? Are some species that are going to be lost from the communities, and in the sense of what does it mean for the overall efficiency of the energy transfer, for example, or grazing effects of fish feeding, and of course development of automatic sampling techniques. I think this is actually quite a substantial challenge, and this would provide a very nice prospect for development. Yeah, that was it.